Look at a monkey and you can tell a lot: the way it moves, the color of its fur, the confidence it carries through the trees. But can you tell how old it is? Not just young or old. Can you actually pin down the years? That question turns out to be one of the more fascinating puzzles in primatology, and the answer has changed dramatically in just the last few years. To understand why this matters, you need to know one thing first: monkeys age. They really age, not just in the vague biological sense. They go gray, their joints stiffen, their vision fades, their social world contracts. They experience something remarkably close to what we call getting old. And for the scientists who study them, knowing exactly how old an individual animal is can unlock answers about disease, evolution, and even human health. So here is how it actually works. From the first weeks of life all the way to the final years. A newborn monkey is surprisingly easy to age. In the first weeks and months of life, the developmental clock is precise and predictable. Researchers have spent decades documenting exactly when specific teeth break through the gum line in different species. In Rhesus macaques, one of the most studied monkey species on the planet, the first deciduous teeth appear within days of birth. The sequence of eruption follows a consistent pattern: canines, molars, each arriving within a known window of time. Up until about 5 years of age, teeth are your most reliable calendar. Field primatologists working in areas with known populations can match a juvenile to a specific birth season simply by examining which teeth have come in and how worn they already are. In the hands of an experienced researcher, this method narrows age estimates down to within a few months. But then the monkey hits adulthood, and that is where things get genuinely complicated. Once all the permanent teeth are in, the eruption method stops working. You are looking at a fully grown animal, and the most obvious age clock has gone silent. In captivity, this is not a problem. Birth records exist. Every monkey in a research colony or a zoo has a file. Researchers know the exact date, sometimes down to the hour. In the wild, it is a different story entirely. A field biologist watching free-ranging macaques in Thailand or capuchin monkeys moving through a Brazilian forest has no records. They have eyes, experience, and a checklist of physical clues that only get you so far. Tooth wear becomes the next tool. As a monkey ages, the enamel on its molars gradually erodes. A young adult has sharp, high-relief tooth surfaces. A middle-aged monkey shows flattening. An old monkey may have significantly worn teeth, and in some cases, tooth loss entirely. Studies of free-ranging rhesus monkeys on Cayo Santiago Island documented that tooth wear and breakage were especially common in older males. The pattern is real and observable, but it gives you a range, not a precise number. You might be looking at a 14-year-old animal, or you might be looking at a 20-year-old one. Without longitudinal records, the gap between those two estimates is nearly impossible to close using teeth alone. The body offers other clues. Muscle mass is one of them. Researchers have identified that iliac muscle mass is one of the strongest predictors of aging-related decline in rhesus macaques. A young adult monkey is dense with muscle. An older animal gradually loses that mass, a process that mirrors what happens in aging humans. Body weight shifts too, often dropping in the later years as the animal struggles to compete for food and maintain the physical effort of daily life. The face tells its own story. In species like the rhesus macaque, the pink facial skin becomes paler and less vibrant with age. Hair condition changes too. The thick, lustrous coat of a prime-age adult gives way to thinning fur. Coat graying, particularly noticeable on the face and extremities, tends to appear in the final stage of life. In rhesus macaques, significant coat graying typically does not appear until an animal is in its mid to late 20s. That matters because rhesus macaques live on a compressed timescale. They age at roughly 2.5 to 3 times the rate of humans. A rhesus monkey in its mid-20s is, physiologically speaking, pushing into what we would recognize as old age. The median captive lifespan sits around 26 years. Animals that make it past 30 are the exception. A handful of individuals, mostly those on caloric restriction in research studies, have been documented living past 40. One wild-type male fed a normal diet is among them, but they are outliers. Across species, the timelines vary considerably. The common marmoset, a small South American primate no bigger than a squirrel, is considered geriatric by 7 or 8 years old. Its entire lifespan typically runs to about 10 years. On the other end, macaques and baboons can live into their late 20s and beyond. Chimpanzees, our closest relatives and technically apes rather than monkeys, have been documented living past 60 in captivity. Now, here is where the behavioral story comes in. Because aging in monkeys is not only written on the body, it reshapes the entire social life of an animal. Studies of Assamese macaques in Thailand, Tibetan macaques in the mountains of China, and Barbary macaques kept in large naturalistic enclosures in France have all found the same consistent pattern. As monkeys age, their social world gets smaller and more deliberate. Old females in particular show a marked reduction in initiating contact with others. They approach fewer individuals. They engage in fewer conflicts. They become, for lack of a better word, choosier. Researchers observing more than 100 Barbary macaques across different age groups found that older animals were also less curious about novel objects placed in their environment. It was not that they had lost all interest in the world around them. It was that their interest had become focused and selective. This is not simply because they have less energy or have become withdrawn in a depressive sense. Researchers at the German Primate Center found that older Assamese macaques in the wild actively and strategically narrowed their social circles toward their most important relationships. The parallel with human behavior is striking enough that scientists have described it as a primate-wide feature of aging, rather than something unique to our species. Cognitively, the picture is also recognizable. Studies at primate research centers have tested aged rhesus macaques on object recognition memory tasks alongside younger animals. The older monkeys showed significant impairment. Their brains, scanned with advanced imaging, showed changes in white matter and some expansion of brain ventricles consistent with tissue loss. Not catastrophic collapse, but a measurable and gradual decline, the kind of slow erosion that any neurologist would recognize. And then science crossed a threshold that changes everything: the epigenetic clock. DNA methylation is a chemical process that tags regions of the genome in patterns that shift predictably over time. The pattern of these tags does not just reflect how old a cell is chronologically, it reflects how fast that cell has been aging biologically. In humans, researchers developed clocks that could read these methylation patterns and produce an age estimate of startling accuracy. Then they built the same tools for monkeys. A 2024 study developed an epigenetic clock for rhesus macaques using over 1,000 blood samples from animals with known ages. The result was a model that predicted chronological age with a correlation of 0.983 and a median error of just over 2 months. That is not a rough estimate. That is a precise, scientific answer to a question that fieldworkers once had no reliable way of answering at all. For wild populations where no birth records exist, researchers have now gone further. A study on wild capuchin monkeys developed an epigenetic clock using DNA extracted from fecal samples, which can be collected without even touching the animal. That clock predicted capuchin age to within about 1.5 years. For an animal with a lifespan of roughly 45 years, that represents an error of around 3.5%. The implications extend beyond satisfying curiosity. Knowing the true biological age of an individual monkey compared to its chronological age tells researchers whether that animal is aging faster or slower than expected. Stress accelerates biological aging. Social isolation does too. High social status in male baboons has been linked to faster epigenetic aging in at least one major study. Early life adversity leaves a measurable mark in the methylation pattern, detectable years later. This is the deeper reason the question of monkey age matters. These animals are our best living window into what aging actually does, why it does it, and what shapes the pace of it. The same biological machinery that grays a macaque's fur and shrinks its social circle and erodes the sharpness of its memory is running in us. On a different schedule, perhaps, but running all the same. The tools have caught up with the questions. And what researchers are finding is that the story of a monkey's life, written in its teeth and its muscles and its fading fur and its DNA, is a story we recognize. It is our story too.